Hi everyone, Felix here. Sorry I couldn't be there in person. I ran into some last minute passport problems, but I'm excited to tell you all about some of the exciting work we've been doing on TRPV3 in terms of both structure and function. So the interest in TRPV3 really stems from Olmsted syndrome, which is a rare genetic disorder that causes severe hyperkeratosis and debilitating, mutilating skin behavior, which is really severe symptoms. And whole genome sequencing of patients with Olmsted syndrome shows 4 gain-of-function mutations in the TRPV3 gene. And so this makes TRPV3 somewhat of a promising target to study when it comes to understanding these hyperkeratotic skin diseases. So here's a simplified diagram of TRPV3 channel, so we can see S1 through S6 helix embedded within the membrane. Most notably, in terms of the 4 gain-of-function mutations that were observed in Olmsted syndrome, we can see that they're located at arginine 416, tryptophan 521, glycine 573, and tryptophan 692. So these were the 4 mutations that were discovered in the genome of patients with Olmsted syndrome. So in terms of our lab, we were able to culture cells to express both wild-type TRPV3 as well as mutant TRPV3, so 4 mutants each at one of these Olmsted mutation sites. And then upon expression of these channels, we were then able to perform patch clamp recording, which is a way of letting us see the amount of current that is passing through these cells. So these TRPV3 channels are at their core essentially ion channels, and so basically when they're open, they allow current through, and so patch clamp is a way that allows us to detect the amount of current that's passing through these channels. So this is patch clamp recording of wild-type TRPV3 from our lab. And so things to note is that body temperature around 37 degrees Celsius, there is no current. So that basically indicates that the channel is closed and inactive. Whereas as you increase the temperature, you can begin to see current. And so that means the channel is open and letting current through. This makes sense because the TRPV3 is temperature activated, so it becomes open upon exposure to heat. So for our first Olmsted mutant TRPV3 phenotype, so this was mutated at arginine 416, we can see from the patch clamp that at baseline body temperature, 37 degrees, that there's already some degree of current passing through these channels. So this indicates that this mutant channel is active at baseline body temperature, which is in contrary to the wild type that you just saw, where at body temperature the channel is closed. So there's some degree of constitutive baseline activity. And here we have our next TRPV3 mutant. So this is mutated at tryptophan 521. And again, we can see from the patch clamp that at baseline 37 degrees body temperature, there's already some current being let in through these channels, which indicates that this channel, similar to the last mutant variant, is open and active at baseline body temperature. And this is different from the wild-type TRPV3 patch clamp you first saw, in that the wild-type appears to be completely closed and inactive at body temperature, whereas these mutant TRPV3 channels appear to be open and active at baseline body temperature. So this indicates that these mutant channels have some level of baseline activity at 37 degrees body temperature, even when not stimulated with additional heat, which is what's normally required to activate the wild type version of these channels. So all the patch clamp recordings you've seen up to this point have been performed on whole cells. So for our next Olmsted TRPV3 mutant, which was mutated at glycine 573, the cells cultured from this variant were actually so unstable that we were unable to grow enough cells to perform whole cell patch clamp. And so instead we salvaged whatever little membrane we could and then performed a patch clamp on just the membrane itself. And so this patch clamp was actually performed at room temperature, and it showed this mutant TRPV3 to be fluctuating between open and closed states, thereby indicating activity at room temperature. And this is particularly striking because TRPV3 is traditionally speaking activated by heat. And so the fact that it is active at room temperature basically indicates that this mutant is constitutively active at baseline without any need for additional temperature requirement. And here we have our last TRPV3 Olmsted mutant variant mutated at tryptophan 692. And again, this patch clamp was taken at room temperature, and it shows that the channel is active and fluctuating between open and closed states. And again, this is particularly striking because these channels normally require heat stimulation at temperatures much greater than body temperature to become active. So the fact that this mutant variant is somewhat active at room temperature, which is much cooler than body temperature, basically suggests that this mutant phenotype is likely constitutively active at baseline in the patients who possess these mutations. So in summary, our functional study with patch clamp showed that these 4 Olmsted TRPV3 mutations produced channels that were constitutively active at either baseline body temperature or, to the latter two, at even room temperature. And so this basically means that these channels are constitutively open without the need of any heat stimulation that would ordinarily be required by the wild type channels to become functionally active. So that's it in terms of functional study. Next, we're going to delve into the structures of these channels. So our lab is able to isolate the TRPV3 channels from the cultured cells and then perform cryo-EM imaging on the channels, thereby allowing us to get atomic level 3D structures of these TRPV3 channels. And so we did this for wild-type TRPV3 as well as one of the Olmsted variants, the arginine 416 mutant variant. And so the image you're seeing here is cryo-EM atomic 3D structure of the wild-type TRPV3 that our lab imaged. And then here's the cryo-EM structure of the mutated arginine 416 Olmsted TRPV3 variant that was, again, isolated and imaged by our lab. And here we're zooming into one of those TRPV3 transmembrane subunits. And this is where the 4 Olmsted mutations can be found. And again, in our cryo-EM mutant structure, we only mutated the arginine 416 location, and we left the other 3 sites alone. And so here we have the cryo-EM structures of both the wild type and the mutant arginine 416 laid on top of each other, and we can see that there's remarkable overlap and similarity between the 2 structures. However, there are slight differences between the structures, which we are now going to delve into detail. So before we delve into the first difference, just to orient you to several important structural landmarks, we have the S2, S3 loop, the trip helix, and the arginine loop. And so these 3 are going to be involved in the first structural difference that we see between wild type and mutant. So on the left, we can see the wild type, and on the right, we can see the mutant. So essentially in the mutant, the trip helix, S2, S3 loop, and the arginine loop are all brought closer to each other. Now for the second structural difference, again, some important landmarks are the S4-5 linker helix, the S6 helix, the trip helix, and then also the arginine loop, which is right adjacent to the site that we mutated, which is arginine 416. So again, the wild type is on the left and the mutated Olmsted variant is on the right. And so we can see in the Olmsted variant that the S4, S5 linker helix, the S6 helix, and the trip helix are actually pushed away from each other. They're spread further apart. In the diagram, it looks like that they're closer to each other on the right, but in fact, when you measure the actual distances, they're further away from each other. And then in terms of the trip helix and the arginine loop in the mutant variant on the right, we can see that they're interacting and closer together than in the wild type on the left. So that's it in terms of the structural comparison. Basically, we compared the wild type cryo-EM structure with the mutated arginine 416 Olmsted variant cryo-EM structure. Now, a third structure we just recently completed is another Olmsted variant, this time at tryptophan 521. And so we just finished constructing the 3D model for that structure, and we're hoping to make comparisons with wild type in the near future. Be sure to follow the PI of this work at his university webpage. And consider following me on LinkedIn and Twitter. I'm no longer active on trip channel work. I am now entirely focused on both artificial intelligence and single cell analysis. terms of artificial intelligence, I'm currently working on large language models, and in terms of single cell, I am analyzing imaging mass cytometry data, which is spatial single cell proteomics.